So dear student, today we are going to discuss the antiviral drugs. By the end of this lecture, you will be able to describe the key characteristic, classification and mechanism of action of these antiviral drugs, and describe the symptoms of the activity of each antiviral drugs, then the pharmacokinetics, the mechanisms, therapeutic uses and clinical uses of these drugs. So to introduce viruses, you might know that viruses consist of nucleic acid surrounded by one or more protein. They lack cell wall and cell membrane, and most viruses contain enzyme essential for viral replication inside the host cells. So basically, they rely on the host cell for their replication. That's why they are called obligate parasites. Viral agents inhibit virus-specific replication events rather than the host cell's directed nucleic acid or protein synthesis. Basically, these viruses are difficult to treat as compared to bacteria because they know they are obligate parasite. They uses the host cells protein or nucleic acid for replication, so they are very difficult to treat as compared to the normal bacterial cells. Along with that, the virus the along with that the symptoms of viral infections come late, while the symptoms of the bacterial infections come gradually. So that suddenly start and gradually increases with the replication of bacteria. But unlike bacterial infections, viral infection, the symptoms of the viral infections comes late when the virus is already multiplied. So that's why it's difficult to treat viral infection as compared to bacterial infection. Now, understanding the viruses, they are different from other microbes that we have described. A virus cannot penetrate, cannot replicate on its own. It must be attached to and enter the host cells, and then it uses the host cell energy to synthesize proteins, DNA, or RNA. Viruses are difficult to kill because they live inside the cells that we have already described, and any drug that kills a virus may also kills the host cells as well. So that's why we have to target only the bacterial or the viral specific proteins in order to have low side effects. This is the replication of the virus. Like virus, first it has to attach the cells, then utilizing then it's uncoated, then enters into the host cells nucleus, and then utilize the messenger RNA or proteins of the host cells, and then replicate, and then releases from the cell and infected other cells of the host. So cell entry, attachment, penetration, uncoating, translation, and transcription and translation of the viral genome, assembly of viron component, and then releases to infect other host cells. So the drug has to utilize this cycle. They either can inhibit the entry of the viruses into the host cells, or they can mutate these or inhibit the replication of viruses within this host cell. So we will discuss different drugs which will inhibit these different steps in this viral cycle. So this diagram shows the mechanisms or the site of action of different drugs that are inhibiting the different steps in the cycle or in the life cycle of the viruses. So like here you can see alpha globulins or gamma globulins. They inhibit the penetration of viruses into the cells. Amantadine, they inhibit the uncoating of the viruses. Purines, pyrimidine analog, they reverse the transcriptase inhibitor, so inhibit the nucleic acid synthesis and then inhibit the viral replication. Similarly, methamezoles also reverse the or inhibit proteases for the viruses, and rifampicin or rifampin uh, block the packaging and assembly of the repackaging and assembly of the viruses, and then inhibit the release. So to inhibit the other cells, the infection of the viruses. So in this lecture we will be discussing these types of antiviral drugs. Viral classification: viruses can be either RNA viruses or DNA viruses, like HIV, hepatitis A, C, mumps, rubella. Influenza viruses, they have RNA, while these hepatitis B, herpes viruses, it's also called cytomegaloviruses, herpes simplex viruses, varicella zoster viruses. These are DNA viruses. So we will mainly focus on the drug. This is only for the introduction of the virus. You might have discussed this in detail in your microbiology classes. Now, classification of the drugs. Now, different drugs are effective against different types of viruses, like in anti-herpes viruses. Herpes viruses, acyclovir, valcyclovir, famciclovir, these are effective against the herpes viruses. Influenza viruses, Amantadine, Ribavirin, oseltamivir, zinamivir, these are effective against the influenza viruses. And then we have anti-hepatitis drugs, which are effective against the hepatitis virus causing the hepatitis, like Ribavirin. These are the types of interferon alpha, pegylated interferon alpha, Ribavirins. More commonly used drugs is anti-hepatitis. Antiretroviruses, which are affecting the HIV, but it's not including in your course. So we will discuss mainly these three types of drugs. Antiviral drugs. Many antiviral drugs are purine or pyrimidine analogs. Many antiviral drugs are prodrug. They must be phosphorylated by the viral or cellular enzyme in order to be effective. So remember the definition of prodrug, which is coming again and again. So prodrugs are those which are not active in their original form, but when they are taken into the body and after biotransformation, they become active by the different enzymes within the body. So mostly these antiviral drugs are prodrugs. So they must be phosphorylated by the viral enzyme. So this is very important. Like if a person has viral infections, then these prodrugs can be converted to its active form. While if a person don't have any viral infections, then these drugs will not be converted to its active form and will be released as such. So that's why those drugs which are metabolized or activated by the viral enzymes, they are safer in terms that they will not be converted to active drugs. So they must be phosphorylated by the viral or cellular enzyme in order to become active. Antiviral agents inhibits active replication, so viral growth resumes after drug removal. Normally they inhibit the replication of the viruses, so if you stop the medication, then the viruses resume their replication. Key characteristics of antiviral drug. So that what we have discussed, they should be able to enter the cell which is infected by the viruses, interfere with viral nucleic acid synthesis and or its regulation of the synthesis. Some drugs interfere with the ability of the viruses to bind the cell, so they will only inhibit the spread or the propagation of these viruses to infect healthy cells. So they will inhibit the healthy cell to be infected by these viruses. They only inhibit the entry of the cells of the viruses into the cell. Some drugs stimulate the body immune system. This is very important for viral infections. If you have a proper or competent immune system, then these viruses can be we can overcome these viral infection. Best response to antiviral drugs in a patient with competent immune system. So a patient he has a competent immune system, if she has a competent immune system, then these drugs respond best to these antiviral infection. A healthy immune system works synergistically with the drugs to eliminate or suppress viral activity. So you remember the definition of synergistic. So we have additive or synergistic or potentiation effect. These are when two drugs are increasing each other effect more than their normal or additive effect. Herpes viruses are associated with the broad spectrum of diseases like coarse or viral encephalitis, genital infections. So these drug will inhibit these viral infection. It was associated with the herpes viruses. The main drug or the prototype drug in this class is the acyclovir, valcyclovir. They are normally a similar drug, famciclovir and penciclovir, gencyclovir. So all they have the cyclovir, mostly phosgrenate, and these are the newer drugs. So we will discuss this in detail in the coming slides. Valcyclovir is a prodrug of acyclovir, so they have similar action. This is a prodrug basically, and this is inactive in its original form. So first it is converted to acyclovir, and then it has the same activity with better bioavailability. So they have better bioavailability. Famciclovir is also a prodrug of penciclovir and has the greatest bioavailability. So their bioavailability is more than the other one. Penciclovir is used only topically, while famciclovir can be administered orally. So pharmacology of acyclovir and related drugs that are related to acyclovir. So acyclovir, valcyclovir, gencyclovir. These are all guanine nucleotides. So this is the guanine, and they are different derivatives of this new guanine. Their unaffected cell do not phosphorylate acyclovir. So this is the advantage of this drug, that this will be only phosphorylated to in the affected cell, because it is phosphorylated by the viral enzyme thymidine kinase. So it is not effective in patients who do not have viral infection, or in uninfected cells it will not be phosphorylated. So only those cells will be in those cells it will be phosphorylated which have viruses in those cells. Now this is the mechanism of action of acyclovir. Acyclovir is converted to its active form, acyclovir triphosphate, with the help of herpes simplex virus thymidine kinase. So this thymidine kinase is produced by the herpes simplex virus. So those drugs which have herpes simplex viruses, they will convert the acyclovir to its active form, acyclovir triphosphate. And then acyclovir triphosphate inhibit the viral DNA polymerase, and thus leads to the inhibition of viral replication. So absence or partial production of viral thymidine kinase is responsible for the drug resistance. Now, how the drug resist? Sometimes these drugs can mutate this thymidine kinase. So the absence of those viruses which do not have thymidine kinase, or if they are partially producing this viral thymidine kinase, then the drug will be ineffective in that condition. Or if there is a mutation in these thymidine kinase, then these drug will become ineffective in those patients. Pharmacokinetics of acyclovir. Oral bioavailability is comparatively lower, around 20 to 30%. However, it can be used topically or it can be given intravenously, and it can distribute in all body tissues, including the CNS. It can cross the blood-brain barrier, and that's why it can have these central nervous system effects. And mostly, it is excreted through the renal route. So these are the therapeutic uses of this antiviral drugs, which are acyclovir, and it's the drugs that are related to acyclovir. And based on their pharmacokinetic activity, they can be used for different types of infections. Like for herpes labialis or genital herpes, acyclovir is a drug of choice. Or for cytomegalovirus of the retinitis, so viral infection of the retina in immune compromised patient, then ganciclovir is the drug of choice. These are the other alternative for the ganciclovir. Along with that, trifluoridines and idoxuridines, they can be used for the treatment of HSV keratoconjunctivitis, that is viral infection of the conjunctiva. Adverse effects: nausea, vomiting, diarrhea, most common side effect associated with most of the drug. Nephrotoxicity is one of the main cause, main side effects, and myelosuppression. So here what happens that the bone marrow activity is decreased. These drugs decreases the bone marrow activity, which results in the fewer blood cells, white blood cells, red blood cells, and the in the platelets, so it leads to neutropenia and thrombocytopenia. Mostly this myelosuppression is associated with ganciclovir. Then in second category, we will discuss the anti-influenza drugs like amantadine, rimantadine, oseltamivir, and zanamivir. So these are anti-influenza. They are effective against the influenza virus. So amantadine and rimantadine, they are effective against the influenza viruses, and their mechanism is they inhibit this M2 protein, which is responsible for the uncoating of the viruses. So if the virus is uncoated, it will inhibit its further replication. So the mechanisms, it's inhibition of viral uncoating by inhibiting the viral membrane M2, which is responsible for the uncoating of this membrane. Amantadine also has this anti-Parkinsonian effect. It is not related to this M2, but number of studies have suggested that it can have this. It improves the motor activity of the in the patient which have Parkinsonism, and that is most related with its dopamine activity. It can increase the concentration of dopamine at the synapse in the brain, and that they markedly improve this motor activity again in Parkinsonian patients. Amantadine, the pharmacokinetics. Amantadine and rimantadine both have anti-influenza effect. They are most commonly the similar. The only difference between them is that amantadine can cross the blood-brain barrier, while rimantadine cannot cross the blood-brain barrier. So this amantadine has more CNS effect, while this rimantadine do not have a central nervous system effect. So pharmacokinetics well absorbed orally, excreted unchanged in the urine. Adverse effects include headache, dizziness, that is mostly associated with crossing this blood-brain barrier, concentrating, difficulty in concentration, seizures, nausea, diarrhea, and dry mouth. So these side effects are common with the rimantadine. But this is mostly these headaches, dizziness, difficulty in concentration, and seizure. It is mostly associated with amantadine, but not rimantadine. So this is the rimantadine. It has the same mechanism of action, same therapeutic use, but it is more potent than amantadine, and does not cross the blood-brain barrier, is opposite to Amantadine, which can cross the blood-brain barrier and reach the central nervous system. So the side effects mostly that's the same, but apart from that central nervous system effect. Now, in this class of antiviral drugs, we will be discussing the anti-influenza agents. So, oseltamivir and zinamivir, they are also called neuroaminidase inhibitor. And what they do basically, they inhibit the neuroaminidase enzyme, which is present, which is essential for the replication of the viruses that causes influenza. So these viruses contains this neuroaminidase, and this neuroaminidase is responsible for the spread down of this viruses. So when we inhibit this neuroaminidase enzyme, it prevents the release of new virons and their spread from cell to cell. So it is used for the prevention and treatment of influenza A and influenza B. This is the mechanism of action. Again, influenza A and influenza B, they have these different types. So it depends on the influenza is most commonly a seasonal infection, while influenza A is also. In this class, we will be discussing the anti-hepatitis agents. So far, we have identified the A, B, C, and D hepatitis viruses, and E along with that E as well. So of this group, hepatitis B and C are the most common cause of this hepatitis, chronic hepatitis, cirrhosis, hepatocellular carcinoma, and are the only hepatic viral infection for which therapy is currently available. So we have currently therapy available for B, hepatitis B and C. The agent included in this class are interferon alpha, ribavirin, Ribavirin, Antiviral. These are also can be used for hepatitis B viruses, and along with that it can be used for the HIV infection as well. So we will be discussing these interferon alpha and ribavirin in this next slide. Interferon, these are endogenous proteins, induce host cell enzymes that inhibit viral RNA translation and cause degradation of viral messenger RNA and transfer RNA. So if you can see here the mechanism of actions, on one hand it inhibits the host cell enzyme, and that inhibits the replication of the viral messenger RNA or transfer RNA. On the other hand, it can bind to this membrane receptors on the cells and inhibit the entry of the viruses into the host cells. So it binds to membrane proteins on the cell surface and may inhibit the viral penetration, and thus inhibiting the uncoating, messenger RNA synthesis and translation, and viron assembly and release. So it is protecting the healthy cells from from penetration of the viruses, and also interfere with the RNA and synthesis of the virus. So they are very much effective in this viral infection. Okay, interferons have many types, like interferon alpha, pegylated interferon alpha. They are used for the treatment of hepatitis B viruses, normally used in combination with ribavirin in the treatment of hepatitis C viruses. Major adverse effects include flu-like symptoms, leukopenia, thrombocytopenia, cardiotoxicity, and impairment of fertility. Pegylated interferon alpha has a longer half-life. The flu-like symptoms are mostly associated are present in the initial stages, or at the start of the treatment, and that include like fever, chills, and gastrointestinal disturbances. Also, it can also cause bone marrow separations, which can cause this leukopenia and thrombocytopenia. Cardiovascular toxicity and impairment of renal perfusion, it's less common. Ribavirin is the second class in this anti-hepatitis virus, anti-hepatitis antiviral drugs. However, it is used in combination with interferon alpha. It is phosphorylated intracellularly in the host cells and inhibit the viral RNA-dependent RNA polymerase, and thus inhibit the replication of DNA and RNA viruses. So that's why it is used in combination with interferon alpha for the treatment of hepatitis C virus infection. It is metabolized in the liver and excreted renally. Major toxicity is hemolytic anemia, because blood cells do not have these phosphorylated enzymes, so that it accumulates in the red blood cells and causes lysis of the red blood cells, which lead to hemolytic anemia. So this is all about the antiviral drugs, where we have discussed the anti-hyperviruses, anti-influenza viruses, and the anti-hepatitis viruses. Acyclovir, valcyclovir, famciclovir belongs to this anti-hyperviruses. Anti-influenza, Amantadine, Ribavirin, and in the anti-hepatitis we have discussed interferon, different types of interferon and ribavirin antiviral agents. So that's all. So thank you for being with me, and if you have any questions, you can email me or you can reach me on my office extension. Thank you.